Okay, fabulous AP Bio 2 kiddos, it's Miss Lynham, and we are starting our next video in Unit 3, Cell Structure and Function, and I believe that we need to start with transport on this one. So we're going to start on slide 34, so get your notes out and your highlighter and something to write with, and get ready to rock and roll, because here we go. We're going to talk about transport of molecules across the cell membrane. Okay, so the topic, first of all, let me get my pen going, rocking and rolling here. We're talking about what can go in and out of the cell. So we already know that the cell membrane is selectively permeable because it can regulate what goes in and what goes out, which helps maintain homeostasis in the cell. So I'm sure you're probably wondering, well, what are some things that can go in and out of the cell, and do those things go through easily, or is it harder to get those through? So let's talk a little bit about the membrane permeability. Okay, we're going to talk about the things that can go through easier than other things. Okay, so membrane permeability. So first of all, the things that go through easiest are things that are nonpolar. Okay, nonpolar. So these go through easiest because if the cell membrane in the interior is hydrophobic, the things that are nonpolar or hydrophobic are going to go through with ease. Okay, so what are some things that you think that we've talked about in this class that would be nonpolar hydrophobic molecules that might go through the cell membrane with a little bit of ease? Okay, hopefully you mentioned some gases like carbon dioxide, oxygen, and then we also talked about hydrocarbons. So because those things are not charged and they're nonpolar, they can dissolve very easily in the cell membrane. Okay, so as they're moving through, they dissolve without any trouble and move right on through. So the example that you would see there is this picture, which we're going to call this one A. A is showing you simple diffusion of carbon dioxide or oxygen through the phospholipid bilayer. Okay, so this could be CO2 or oxygen, and you'll notice it's simply going through the phospholipid bilayer, and there's no need for any protein molecule like you'll see in picture B. In picture C, these use proteins that span through the membrane. So picture A is an example of passive transport. Okay, passive. How can we tell that? Passive transport is defined as movement of substances from a high concentration to a low concentration with the concentration gradient. So if you look at the circles, there are, I don't even know, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, like 16 solute molecules on this side of the cell membrane, 2 solute molecules on this side, and the arrow is showing you that it's going from 16 down to 2. So that is definitely movement from a high 16 to a low concentration, which is 2. So picture A is definitely an example of passive transport. Now, because there's not a protein involved, I can tell you specifically, picture A, the example represents diffusion. Diffusion of carbon dioxide and oxygen simply through that phospholipid bilayer. Okay, picture B is still showing you passive transport, because look at the triangles. I'm not going to count them, but there are a lot more triangles on this side compared to the 3 that are on this side. So this is also going from a high to a low concentration. So picture B also represents passive transport, but you'll notice it uses a protein molecule. So this one would be something known as facilitated diffusion. And facilitated diffusion is defined as diffusion from high concentration to a low concentration, but needing to use a protein molecule to get that through. And this one is actually a protein channel, whereas in picture C, it's also going high to low. So this would also be passive transport, and it's another example of facilitated diffusion, but you can see the difference in the shapes of these, because some are built specifically on size to fit those things through, and then others serve as little channels that can allow stuff to go through. Picture B would represent something called an Aquaporin, which is designed to allow water to go through a little channel. Picture C might represent a glucose channel, or a glucose protein, so it's allowing sugar molecules that are larger, like glucose, fructose, and galactose, to go through, and these would be specifically designed, these proteins, to fit the proper sugar that would need to move through. So in this picture right here, this first picture that you see, the 3 different models, everything here is named passive transport. Okay, passive transport on this side. One more time, how do we know it's passive? Because it's moving from a high concentration to a low concentration. And the first diagram represents what moves through easiest. Okay, in this first picture, these are non-polar hydrophobic substances such as carbon dioxide and oxygen and other hydrocarbons that will dissolve through that phospholipid bilayer. These other two pictures, they'll go a little bit slower. Okay, the movement will go slower, but it's obviously sped up by a protein compared to what it would if there wasn't a protein. But some things that go through that would be water through an aquaporin, or glucose, fructose, to lactose. Ions could also go through one of these. But I did want to mention that water can also slip through the phospholipids despite the fact that it's polar, because it's not a charged particle. So even though it's polar, it can slip through these phospholipids, but it's a lot slower than it would if it would use an aquaporin to go through this water channel. If it went through that, it would be osmosis, which is one more example of passive transport. Okay? So as far as the permeability of this membrane, we talked about things that go through easily, right here. Things that would have a harder time getting through would be things that are polar, like glucose is a polar molecule, and that would need to go through a specific protein. Just like we said, water goes faster through an aquaporin, and then if it's a very polar molecule, it's going to, like I said, need to go through one of these proteins. Okay, so that's kind of the movement in that regard. Okay, the second picture over here is going to represent active, which is the second type of transport, active transport, which is defined as going from a low to a high concentration against the concentration gradient, against the gradient. Okay, so in this picture, these little squares, you can see that there are only two on this side, and they're moving through where there are 1, 2, 3, 4, 5 on this side. But do you see this little yellow thing? This is representing the use of energy. So to go from a low concentration to a high concentration is going to require ATP. Passive transport, no energy is needed. Okay, no energy needed. The reason being, guys, is things want to go the path of least resistance. So if you're thinking about a skateboarder, let me just draw my little picture that I always draw. If you're thinking about a skateboarder up here, and he's on the top of the hill, here he is on a skateboard, it's a lot easier for him to go down that hill than it is for him to be on the bottom and have to climb that hill. Just think of the time when you were a toddler and you went down a slide. Going down the slide is really easy. Okay, going down that slide, that's the concentration gradient. It represents potential energy. So to go from high to low, my pen is working really slow right now. Okay, so to go from high to low, that was a epic failure right there. Here's my hill. Here's my skateboarder at the top of the hill. Here he is. To go from a high part of the hill to the lowest part of the hill is not going to require much energy. So going from a high concentration in the cell to a low concentration, there is no energy needed. But to go from low to high, you would have to go against the gradient, against the flow of things. The natural tendency is to go from high to low. So if you would go from low to high, you're going against the flow. So that would require energy, which is why the two types of transport, we call them passive and active. Okay, the two types are passive and active. So passive is no energy needed, active is energy needed. Passive goes high to low with the flow, active goes low to high. Okay? All right. So now let's get into some specifics about passive transport. Okay, I'll let you kind of look at this diagram because it's cool. This little animation is showing you the movement of molecules across the cell membrane, and you can see that the arrows get really small and stop the net flow right there once it reaches equilibrium on both sides. So passive transport, let's go ahead and define it. If you have your highlighter or something to write with, go ahead and highlight the term passive transport. That's a vocabulary term. So it's movement of materials. Okay, this could be, by the way, solute molecules. It doesn't just have to be in a cell. You could actually talk about the diffusion of perfume particles or odors about a room, and that would also diffuse. But it's movement of materials, because we're talking about the cell, from a high to a low concentration with no energy output required. The first example, okay, grab your highlighter, diffusion, simple diffusion. This is random movement across a membrane down the concentration gradient, and this would be a substance, like we said a minute ago, such as oxygen, carbon dioxide, or hydrocarbons. These substances would dissolve easily in the phospholipid bilayer and move right across, and once it reaches equilibrium on both sides of the cell, the net movement would cease. Net movement means overall movement will stop, but you will still have equal rate of exchange. For some particles that went to this side, some would go to that side. But the net flow, the net movement, once equilibrium is reached, would go ahead and stop. Okay, so diffusion. Another example, like if you were doing a non-cell membrane example, is you could get a beaker, put some water in there, and if you dropped ink or food coloring into that beaker and you dropped it right here, that drop would spread out, and eventually that beaker would become all one color because that ink would diffuse from a high concentration to a low concentration until it reached equilibrium inside the beaker. So that's like a non-cellular example. When we're talking about the cell, this might be the movement, let's say, of a lot of oxygen on this side, and it would diffuse across until you had equal oxygen on both sides, at which point the overall movement would cease at that point. Okay? So that's diffusion. You can see the animation one more time. It's kind of cool. So high to low with the flow, no energy needed. Examples are oxygen and carbon dioxide diffusing right through the membrane until equilibrium is reached. Okay, here's another picture. This one is actually from Time magazine. So in the first scenario, you can see, look at all of these blue solute molecules. Let's say this is oxygen. So in picture number 2, if all of these oxygens are on the outside of the cell, they will start diffusing through. So now you can see 1, 2, 3 here, and there's a couple that are dissolving in the phospholipid bilayer. And then in picture 3, we've reached equilibrium on both sides of the cell membrane. So net movement will have ceased at that point. Okay, here's another picture where the top model shows you the diffusion of one solute. So you see all these little yellow dots on the left side. Okay, those will diffuse to the right side until you see in the last picture you've reached equilibrium. Okay, the bottom scenario is showing you maybe 2 solutes at one time. So let's just pretend like the purple is oxygen, the yellow is carbon dioxide, so there's a high concentration of oxygen on the left, a high concentration of carbon dioxide on the right, so oxygen would diffuse to the right, and carbon dioxide would diffuse to the left. So in picture 2, you can kind of see some of the yellows or oranges went to this side, some of the purples went to this side, and in the last picture, you have an equilibrium that has been reached, because you've got equal solute concentration of both of those on either side of the cell membrane. Okay? All right, next example, we're going to talk about facilitated diffusion. And like I said earlier, facilitated diffusion, this word facilitate means to help. It's like to help out, to initiate. So this is still going to be a type of passive transport. So you might want to go ahead and write here, we're still going high to low with the flow, and I'm going to put with the concentration gradient, but unlike simple diffusion, this is going to need the help of a transport protein. So we're going to need proteins, so I'm going to circle this right here, we're going to need proteins, because these must be utilized by larger molecules, some that are charged, okay, let's say an ion that would need to get across, a charged particle is one that has gained or lost an electron, or polar molecules such as glucose or water, things that are polar. So these things, due to the fact that they are either charged, they're larger, okay, the larger might be a larger, let's say a protein molecule or an amino acid that's a little bit bigger, an AA, I'm going to go ahead and write that down. Things that are larger, charged, or polar are going to be helped across much faster by the use of a transport protein that is embedded in the cell membrane. So in the picture that you see right here, this one is a specific type. This is called an aquaporin. These are amazing, guys. The rate at which these things allow water into the cell is unbelievable. It's a special water channel. Okay, if my this pen is going to drive me crazy. It's a special water channel that allows water to go through the cell membrane. I will just tell you that they've done a lot of research on these aquaporins, and they say that the research has shown that you can fit 10 water molecules through one aquaporin at a time, which speeds up the rate at which water will enter into a cell. They've actually done a calculation, and they said that by going through 10 at a time, this allows up to 3 billion water molecules per second to pass across the cell membrane. It's unbelievable how fast that can happen. They've actually done studies on our red blood cells, because red blood cells have a high concentration of these aquaporins, as well as kidney cells. And if you think about the job of the kidneys, it makes sense why the kidneys would have quite a few aquaporins to remove like the excess water that we have in our bodies. So these are special little channels that are kind of cool. One little side note that I thought I would write here is they have actually studied some inherited genetic disorders or inherited diseases that do affect transport proteins. There's one in particular that's called cystinuria. It's cystinuria, that if the person is missing the carrier protein that is needed to transport the amino acid called cysteine, you probably remember that one because that's the one that has the sulfhydryl side chain on the R groups, but this results in the formation of kidney stones. So if you know someone who has quite a few kidney stones, I will tell you that it is genetic, and the person is missing a few of these transport proteins that is responsible for getting this amino acid through into the cell. So it's kind of interesting that they've done some research on these. I just thought that was kind of cool. Okay, so moving right along. This is a pretty cool graphic that I found that represents facilitated diffusion. If you look at it, you can see the different movement of these solute molecules across the cell membrane. So in picture 1, you see there are 1, 2, 3, 4, 5 squares, and so that's the highest concentration as compared to the inside of the cell. So those will start moving through these carrier proteins to go to the other side. So in the third picture, you can see that some of the squares have moved into the cell. Now, one thing I didn't mention earlier, I thought now would be an appropriate time to tell you that these proteins, okay, we talk about how they're channels. They're actually in a category called channel proteins. The reason I'm mentioning that is because when we get in a minute to active transport, okay, I'm just going to write it down here, but when we get to active transport, the ones that require proteins are actually called carrier proteins. So there are 2 types of proteins that are embedded in the cell membrane that help with transport. The ones that help with facilitated diffusion, which is passive, are these that are called channel proteins. It kind of makes sense because it looks like a channel is cut out in the middle of that protein, and it's a channel that's specifically designed to fit a certain solute molecule that's going to move across. So if this was glucose, this channel protein would be fit for glucose to move that across. Okay, so like I said, in a minute, when we get to active transport, we're going to talk about some pumping, and the pumps are other proteins that are carrier proteins. I just wanted to make sure I mentioned that now before I get to the next topic. Okay, here we go. By far, this is probably one of the most difficult concepts to understand in passive transport. So it's the third example of passive transport, and it is osmosis. Okay? So just to revisit, the first example of passive transport was diffusion. The second example was facilitated diffusion. And now the third example, I'm just going to put numbers by this, number 3 is osmosis. It is literally defined as the diffusion, so we're still diffusing, and remember, we're still going high to low with the flow, no energy needed. So it's the diffusion specifically, let me change to red, of water. So it's the diffusion of water across a membrane, okay, from one side to the other. This is not using an aquaporin. This one is actually going through the phospholipid bilayer, okay? So there's a word that describes the tendency of a cell to gain or lose water, which is known as tonicity. Okay, so tonicity refers to the tendency of a cell to either lose water or gain water based on this philosophy that water, okay, ready? Water moves from a high water concentration to a low water concentration. That's the way that water has a natural tendency to move. In fact, it creates something known as osmotic pressure because of the fact that it wants to go from a high to a low concentration. Okay, so scientists have created some terms, you see them listed right here, which describe the tonicity of a cell as being isotonic, hypertonic, or hypotonic. And these are comparative terms. What I mean by that is if you have a cell, you can compare the inside of the cell compared to the environment around on the outside of the cell. And so we can compare the internal environment to the external environment, and we can use terms like isotonic, hypertonic, and hypotonic. So these are relative to the cell and the outside environment, okay? So let's define these really quickly. Okay, the first one, I know you guys have looked at your root words. Iso means same, okay? So iso means same. So that would mean that the concentration on the inside of the cell would be the same on the outside of the cell relative to solute, okay? So this means solute concentration, that means how many particles, like sugars, salts, things like those, how much is on the outside of the cell and how much is on the inside of the cell are the same compared to each other. So if that were to be true, there would be no net movement of water. That means water would not enter the cell, water would not leave the cell. So the tonicity is one where the cell will remain the same. You'll have an equal movement across both sides of the membrane. Okay, the hypertonic, I know you guys have heard of somebody being hyperactive, right? If somebody's hyperactive, it's usually because they ate a lot of sugar, and therefore they're like hyperactivity, whereas this is not hyperactivity, it's hyper, which means increase, in tonic, which refers to the solute, okay? We're talking about a lot of salt, or a lot of sugar, or a lot of particles, whatever those solutes are. So I'm going to actually write it above this. Solute could refer to salt, sugar. It's anything that is dissolved in water. So if it's hypertonic, what we say is it's high solute, and if it's high in solute, it's going to be less or lower in water compared to the other side of the cell. Okay, so if it's a hypertonic environment, it's high solute, less water. Just like hypo means under or lower, that would be less solute. Okay, look, less solute. So I'm going to go ahead and write right here, less solute, and therefore it would be higher in water concentration. So just to give you an example, if my pen will serve me correctly, and if it works, okay, I'm going to draw a beaker right here, and I'm going to drop a cell in the beaker. If I told you that inside of the cell was 70% H2O and 30% solute, well, if I dropped it into this beaker and I put inside this beaker 90% saltwater solution, okay, well the salt, 90% salt, means that it's only about 10% H2O. So if I compare the inside of the cell to the outside of the cell, 90% salt, that's more solute compared to 30% salt. So my beaker, in this case, my beaker is hypertonic. Okay, hypertonic. That means more solute, less H2O. My cell would be hypotonic. Okay, the cell would be hypo because it would have less solute and more water. So in this case, the direction of water movement, okay, water moves by osmosis. My dog's saying hi to you right now. Water moves by osmosis from a high water concentration to a low water concentration. So if the inside of the cell has 70% water compared to the outside of the cell, which only has 10% water, water will leave the cell because it goes from the 70 out to the 10. So it goes from high to low. Hang on, my dog is not. Okay, let's try another one. Here's a beaker. Here's my cell. Internal contents, 70% H2O, 30% solute. And I put it into a beaker that has 90% H2O, 10% solute. Okay, which of the two concentrations or contents is the beaker or the cell hypotonic? Which one is hypotonic? Can you answer? Okay, first thing you have to do is break down what does hypotonic mean. That means less, okay, refers to the solute. That means higher in water concentration. So the least amount of solute in this case is going to be the beaker. Okay, so the beaker itself is hypotonic. So my answer on that one would be beaker. Okay, what if I asked you in which direction will water move? Will water go into the cell or out of the cell? Okay, hopefully you answered into the cell because 90% water is higher than 70% water, and water moves from high to low, so it's going to go from the beaker into the cell will gain water, okay? So hypotonic means less solute, higher in water. Hypertonic means high solute, low water, and then iso means equal water to solute concentration on the inside of the cell to the outside of the cell, okay? Now I'd like to show you this bottom diagram. This is actually called a, listen carefully, I'm not exaggerating, this is called a U-tube, but it's spelled like this, U-tube. Okay, not Y-O-U-T-U-B-E, but this is a true U-tube that you can use in experiments. And right down here, there is a structure that represents a selectively permeable membrane. So this serves as an artificial model you can use in the classroom, where there's something across right there that represents a selectively permeable membrane. So in this case, if I told you guys that this cell membrane was not permeable to the solute, okay, so this solute could not go across, the only thing that is permeable is water, what you would be able to do is you could fill in this side, let's say this was side A, I could pour this solute concentration, and then inside B, I could pour a different solute concentration. They would meet in the middle, but because this side, side B, look at my term, is hypertonic, meaning more solute. This side is hypotonic, meaning less solute. If it's less solute, it's high in water. Water will move from high to low, and so water will actually move from side A into side B. And if you notice, this is the after, this is at the end, my water level actually increases inside B. The increase in water is because water moved from a high concentration to a low concentration until both sides are now at equilibrium. So this solute to water concentration is isotonic to this one. When will it stop moving? When it's reached that isotonic, so my net movement will stop, but I will still have an equal rate of exchange across that membrane. So it's a cool little thing that you can use in the science classroom to illustrate the movement of water through osmosis. It's kind of a cool little tool. Okay, this picture is the same thing. So in this case, it's just a different graphic representation. But you can see, here's the selectively permeable membrane. I'm drawing it in green. That's the cell membrane. And it is permeable to water, but not sugar. So sugar is not allowed. So it tells you right here in this diagram, it's an artificial model. It tells you that water can pass, but sugar cannot. So if I start off like this in my first picture, and you can kind of see that the concentrated sugar solution is on the left. So this side would be hypertonic. Okay, hyper on the left. This side is hypo, and so hypo means less solute, high in water. The water is blue. So there's a lot of water on this side. So after a while, once you let these two solutions be near each other, water will go from side A, if this was side A, this was side B, water would go from side A towards side B until it reached equilibrium. And so now my water level increased on this side to allow them to reach an isotonic situation. Okay, does that make sense? Okay, so now for the part that is hard is we're going to talk about the ever so famous water potential. Okay, so our next topic is we're going to talk about water potential. And this is actually what our lab is on. This week we're doing the diffusion osmosis lab, and one of the aspects is we're going to be using potato cores, and potatoes are plants. And you're going to be doing a lab with different molarity concentrations of sucrose and figuring out which potato was placed in each different solution based on what happens to the potatoes, the masses of them, over 2 days. So it's going to be kind of a cool lab, but water potential is it's a method that is used to predict which way water diffuses through plant tissues. Okay, so I'm going to add in right here, this is through plant tissues. Because you know that we don't have a cell wall around the outside of our cell, so when our cell takes on water, it's going to expand a little bit, or our cell can lose water and shrivel a little bit. But when you're looking at a plant with a cell wall around the outside, they're going to have a different mechanism. It's kind of cool because they can have this thing called Turgor pressure that we'll talk about in a little bit. But we can use this process to figure out water potential. Now, this is a kind of a mathematical thing, so don't let it scare you. But for water potential, there is a formula. Okay, put a star right here. This is the formula for water potential, and this looks like a little pitchfork, but this is the Greek letter psi. And so this little Greek letter psi right here, this is the symbol for water potential. So this would say water potential is equal to, this is the potential or the pressure potential, that's what the P is, and the S is the solute potential. So this reads as, and I'm going to put a little W right here, this reads as water potential is equal to the pressure potential plus the solute potential. And once you use the formula, you also take into account this little bullet, which is that water always moves from an area of high water potential. Now I said concentration, okay, that's high concentration, but what we say is if it's a higher concentration of water, there's a higher potential of that water to move. And so water always moves from an area of high water potential to an area of low water potential, or I like to say area of high water concentration to an area of low water concentration. Okay, so we also like to say that it moves from an area of that high free energy, okay, high free energy. So it moves from an area of high free energy to an area of low free energy or low water potential, and so this formula can measure the potential of water to move from high to low. Okay, now the formula, I will give this to you. You do not have to memorize this, but in the formula where it says water potential is equal to the pressure potential plus the solute potential, there's a little bit of configuration here because the solute potential is equal to negative I CRT, where I is the ionization constant. What that means is whatever you're putting into the water, is there a tendency for it to ionize? Well, if salt is used, salt can ionize into an Na ion, Na+, and a Cl negative ion, so salt has an ionization factor, an ionization constant of 2, because it can break up into 2 ions. Okay, so that would be an ionization constant of 2, whereas C is the molar concentration. So if we were using like a 0.6 molar sucrose solution, then the C would be 0.6 in this case. Okay, the R is known as a pressure constant. This is a constant that's given to you. And so R is equal to 0.0831 liter bars per mole Kelvin. And then the T is temperature, which is in Kelvin. And so Kelvin, the formula is 273 degrees plus Celsius, or it's 273 plus Celsius, whatever the degrees Celsius are. And so that's going to be those things, negative I times CRT, that's going to be equal to your solute potential. So once you figure that out, the solute potential plus the pressure potential is equal. That is what water potential is equal to. Okay, so there's a couple things that can impact the potential of water. One thing is if you increase the solute. If you increase the solute, it's lowering the water potential. If you think about it, it makes sense, because if you increase the solute, you're making that solution hypertonic. Well, if it's hypertonic, it's high in solute, it's lower in water compared to the other side. So the water potential is lower as opposed to one that has a very decreased solute. If you decrease the solute, you increase the potential of water. So that makes sense. Okay, now most things that we're going to be considering are in an open container. If it's in an open beaker, the pressure is 0. So atmospheric pressure is always going to be equal to 0 unless I tell you that it's in a closed container or there's some kind of pressure that's being exerted. Okay, if we do increase the positive pressure, if we increase the pressure, it is going to raise the pressure potential, which in turn raises the water potential. Because if I raise the pressure potential, I'm raising the water potential, which means raising the ability of water to move from that solution to another solution. Okay? So let's do a little practice. I have a few little basic ones, and then we'll get into one that's a little bit harder. So here's a little practice problem, and it says, determine the water potential of a cell if the pressure potential is equal to 0.3 and the solute potential is equal to negative 0.5. So all you're doing is using your formula. Water potential is equal to pressure is 0.3 plus negative 0.5. I add those together, so my water potential is equal to negative 0.2. Okay, so that would be my answer, negative 0.2. Not bad, right? Okay, the next one says, calculate the pressure potential if I give you water potential is 0. Well, if water potential is equal to the pressure potential plus the solute potential, and I'm telling you 0 is the water potential, is equal to, I'm asking for pressure, so here's what I'm looking for, plus my solute potential is negative 0.2. So all I do is a little bit of math, and now I know my pressure potential is equal to, I'm going to add, okay, my 0.2 here, I get 0.2 here, so my pressure potential is equal to 0.2. Okay, does that make sense? Alright, so now let's step it up a bit where we do a problem where we can actually look at some molarity and use that ionization constant. Okay, so this scenario is telling you a dialysis bag is filled with sucrose solution, so I would right away draw a little beaker. Here's my dialysis bag. So the bag is filled with a sucrose solution inside the bag of an unknown concentration. We don't know what the molarity is. It's placed into this beaker that we know in the beaker is 0.6 molar sucrose. And it says the initial mass of the bag is it weighs 24 grams. And then after this experiment, it's going to weigh 22 grams. So if my mass decreased, what happened? Okay, hopefully you can say we lost water. If the mass went from 24 to 22, that meant water left the cell. Well, if water's leaving, that gives us a little bit of idea about what's going on because water goes from a high water concentration to a low water concentration or water potential. So that meant that, if you look at the first question, what does that tell you about the molarity of the unknown solution in the dialysis bag? Okay, well, if water's going to leave and go from there out, that would mean that inside the bag was hypotonic. Hypotonic means less solute, more water. So I know that inside that dialysis bag would have to be less than 0.6 molar. So I know it's less solute than 0.6 molar. Okay, so I'm going to put less than 0.6 molar in the bag. Okay, initially is the unknown solution, meaning inside that dialysis bag, would it be hyper, hypo, or iso relative to the beaker? Well, we already broke it apart. We said it had less solute in the bag, so our answer is hypotonic. Okay, now for the part of calculating the initial water potential of the sucrose solution in the beaker, we're looking at the beaker. So we're going to go water potential is equal to pressure potential plus solute potential. Okay, and I know that this is an open beaker, so I'm going to have that is equal to 0 because it's an open beaker. It doesn't say that there's a lid on it or anything. Plus my solute potential, and my solute potential I need to figure out is negative ICRT. So I have to figure that part out, so I'm going to come over here. Negative I, my ionization constant, well, this is sucrose. Sucrose doesn't ionize, so it's going to be negative 1 times C is my molar concentration, so I'm going to multiply this times 0.6 times R. Okay, remember R is our pressure constant, which is 0.0831, and then times my temperature. What tells you temperature is 28 degrees Celsius, so I need 273 plus 28. And so now all I have to do is do my work. So once I do that work, I should get the water potential is equal to 0 plus whatever this answer is. That answer goes right there, and then I'll get my final answer. And you use your calculator, you can do those order of operations, and then figure out your answer right here. If you hold on, I'll do it for you. Let me just push pause. Okay, so I did it for you. I had to pause for a minute so I could get my calculator. My answer, I got negative 15.01. That's an 01. Is that what you guys got? Hopefully you did, and I did it right. Woohoo! Okay, so we're almost done. Hang in there. Bozeman has a really cool link if you want to watch Bozeman video. He also does a video on water potential, so if you want to watch that, you can. And we are almost done. hang in there. I literally think I have 2 slides left because I'm going to do active transport on a different video. So here we go, peeps. All right, this slide kind of talks about osmoregulation, which is, if you think about it, if water is going to go from a high potential or high concentration to a low concentration, that means if you put cells in water in a very aqueous environment, there's going to be a natural tendency for water to go from high to low, regardless of whether you want it to or not. And so some living things have to have a way to regulate the force or the osmotic pressure that exists. Okay, so because water is going to go from a high potential to a low potential, some organisms must have a way to counteract osmotic pressure, or else they literally could burst or not be able to maintain homeostasis. So some cells that have these mechanisms to prevent excess loss or uptake of water, one example is if they have a cell wall. So if it's a bacteria, a fungus, a plant, or some protist, okay, fungus, plant, or some protist, the fact that they have a cell wall physically prevents the cell from exploding or from shriveling up, because the cell wall is a rigid structure. Remember, if it's made up of peptidoglycan or polysaccharide material, or if it's made up of chitin or cellulose, it's a very structurally sound material, and so that cell wall physically prevents the cell from exploding or shriveling up into nothingness. And so that cell wall is one way that some organisms can counteract the flow of water from a high water potential to a low water potential. The second example is called a contractile vacuole. These would be in single-celled protists, okay, such as a Paramecium. A Paramecium, if you remember correctly, is found in pond water. Well, since it's in pond water, if you put that little dude in Lake Houston or a lake or a pond, water is going to want to go into the little Paramecium's body, and if they don't have a way to counteract that force, they would blow up. And so within little protists, they have a vacuole. I'm drawing it right now. It's called a contractile vacuole. So as water comes into their little bodies, water can be pumped out of the contractile vacuole, which is a way that they maintain homeostasis without exploding or lysing. So if it's a plant, fungi, or bacteria, the cell wall counteracts osmotic pressure. If it's a single-celled protist, the contractile vacuole helps counteract osmotic pressure. If it's us, I'll just put humans down here. Okay, if it's humans, we also have a way to counteract osmotic pressure, and it's because our cells are bathed in blood, and blood is not 100% water. Blood is basically 70% water, 30% solute, which is the same as it is on the inside of our cells. And so since we have blood which has solutes or dissolved proteins in it, that helps us counteract osmotic pressure as well. Okay? So let's just say that you did put a plant in an environment that was hypertonic, H-Y-P-E-R. So you exposed a plant to a lot of salt. Okay, well, if you put them in a lot of salt, it's a hypertonic environment, what the cell will do is called plasmolysis. It's known as plasmolyzing. It's when they will lose excessive water, and it will cause the cell membrane to pull away from the cell. So I wanted to show you these two pictures. In this top picture, let's just call this one A, right here, and this one is B. What you see is these are red onion cells under the microscope. So I'm going to use black and outline the cell wall. Here's the cell wall, right here. So it's a rigid structure, and you can see the cell wall is shared with several plant cells. And so it's a rigid structure, but this would be a plant cell if it was in a hypotonic environment. So there's a lot of water inside the plant cell. Okay, so it was in a hypotonic situation, so water was in high potential and flowed on the inside. So the vacuole would be really, really big, like this, and push up against the cell membrane, and the cell membrane is nice and tight up against the cell wall. Picture B is the opposite. If you were to put this plant cell in a hypertonic environment, okay, hypertonic means a lot of solutes on the outside, it's like salt water. Water would tend to leave the plant cell, so the water potential inside the plant cell would be higher. Water would leave, but you'll notice the cell wall still stays here. It's still structurally sound. But what has happened is, I'm going to draw, this is the cell membrane right here. So the cell membrane has sucked in, but the cell wall physically prevents the cell from shriveling up. but you'll notice that the water, because it's left the central vacuole, has pulled that cell membrane in. Okay, so that's what you're seeing in that picture. In this picture over here, I love this picture, you're seeing, here's the contractile vacuole of a paramecium. So this one would be open, pumping out a lot of water, and this one is the contractile vacuole right here as well. Okay, so that one's not pumping out as much because water's coming in. It's kind of cool. Okay? Alright, I think the last slide is one where you can see the differences between putting an animal cell and a plant cell in a hypotonic, isotonic, or hypertonic solution and what some of the effects are. So I'm just going to write a few notes on here, and like I said, this is our last slide for passive transport. Okay, so hypotonic, I'm going to go ahead and write over here. This means low solute, high H2O. So if we put an animal cell, okay, notice over here, these are animal cells on this side, the yellow. If we put your cell or my cell in a hypotonic solution, that means a lot of water, water is going to come into our cell and it is going to do something called lysing. To lyse means to split, okay, split or rupture. And so I make a sad face on here. This is not an ideal situation for your cells or my cells, because we do not want to have cells splitting or lysing. That's not a good scenario, okay? But look at a plant cell. Here's the plant cell row right across here. If you put a plant cell in a hypotonic solution, so this is low solute, high H2O, water goes into that plant cell. They are happy, 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 because water goes in, fills up their central vacuole. There is now a pressure against the cell wall, which is known as Turgor pressure. Turgor pressure is a back pressure, which actually prevents more water from entering. So once it reaches that turgor, then it's going to stop taking on excessive water. But what we say is the plant cell is known as turgid because it's nice and plump, full of water. This would be like the happiest the plant is. It would be like the crispiest of celery that you bit into and it was nice and crunchy. So it's a full plant of water. It's known as a turgid plant. Okay, so animal cells, no, 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 don't like hypotonic situations. Plant cells like it. Okay, the second solution is isotonic. This means equal rate of exchange on the environment compared to the cell. So for us, I'm going to make a happy face. This is normal. We prefer to be in an isotonic situation. This would be like 70-30 on the outside and 70-30 on the inside. Okay, so if I had 70% water, 30% solute on the outside, and I had the same thing on the inside, I would have an equal rate of exchange. Water coming in and water going out at an equal rate. That's a happy face. That's normal for animals. Okay, down here, a plant. This is not their ideal situation, so I'm going to make a face that's kind of like this. Not sad, not like totally happy, just kind of existing. It's known as a flaccid plant. It's one that is kind of a little bit wilted. It's like a plant that would prefer to have a little bit more water, but it's still okay, because you would have some water coming in, but they would also be losing some water, so the cell wall still stays rigid. Notice the cell wall is still the same, but it's not as plump as it is if you were to put a plant in a hypotonic solution. Okay, and the last scenario, hypertonic, this would be high solute, low H2O. This is us. That would mean water would leave the cell. That's known as crenating or shriveling. Okay, the cell will shrivel or crenate, and that's not happy for us. That's a sad face. And then a plant cell does the same thing. This is not happy for them. They will do what is called plasmolysis or plasmolyze. That's what I showed you on the picture before. So the cell wall still remains intact, but here's the cell membrane pulling in, plasmolyzing as water has left that plant cell. Okay? So what's ideal for a plant? Ideal is a hypotonic environment right over here. What's ideal for you and me and animals is an isotonic situation. So what's good for a plant is not necessarily good for us. Okay? Alright, that concludes passive transport. Have a great evening.